now our next speaker is Daniela Felix Lopez. She's going to be remote in this talk. So she's going to talk about local adaptation of highly migratory shark in the northern Mexican Pacific. So we're going to see her presentation. At the end, we are going to have time for questions.
Hello, my name is Daniela Felix, and I am a PhD student at the University of Manchester. This project was developed during my master's degree in marine ecology at CITES under the supervision of Dr. Rocha Olivares and Dr. Nancy Saavedra Sotelo, where we looked at the population structure of a migratory shark. Understanding the potential mechanisms that promote population isolation is necessary for evaluating genetic structure patterns in marine populations. But species with highly migratory potential typically exhibit panmixia across regions. However, mechanisms such as environmental factors like distance between ocean basins or environmental heterogeneity promote genetic differences in populations. On the other hand, behaviors such as site fidelity and philopatry drive population isolation in the absence of physical barriers. Migration is primarily associated with reproductive and/or feeding behavior. Males promote with gene flow, whereas females promote genetic structure to migrate to parturition sites. Identifying migration patterns and mechanisms that promote shark population isolation will allow us to identify the population segments that are most vulnerable to fishing extraction. The northern Mexican Pacific is characterized by an environmental heterogeneity displayed between the Gulf of California and the western coast of the northern Mexican Pacific. This oceanographic heterogeneity has been suggested to exert selective pressure on some species, leading to ecological adaptations regardless of the migratory capacity of the species, resulting in the genetic structure and rising speciation processes in many elasmobranchs. In recent years, SNPs have become the most used molecular marker to evaluate patterns of diversity and genetic structure, but also the selective pressure. The smooth hammerhead shark is a semi-oceanic species which is highly migratory and widely distributed with potentially philopatry behavior. It has been catalogued as vulnerable by the IUCN and included in the Appendix II of CITES controlling its exportation, and its capture is regulated in Mexico by NOM 029 test 2006. Recent studies have classified it as vulnerable due to the low abundance in catches during the last 5 decades. But there are only a few studies of genetic diversity and structure which could help assess the population's status and, as a result, better understand its vulnerability to fishing. In the present study, we evaluated the structural patterns of the smooth hammerhead shark Sphyrna zygaena from the northern Mexican Pacific. For this, we used 1,480 high throughput genotyping in 92 individuals collected in 4 regions in the northern Mexican Pacific between 2014 to 17, and were divided in purely neutral and used for genetic structure analysis and putatively adapted to test selective pressure patterns. In the case of purely neutral, according to our results, no pattern of genetic structure emerged from the analysis of 1,456 neutral SNPs. It means the northern Mexican Pacific contains only 1 genetic unit. Although the Bayesian cluster analysis indicated the possible existence of 3 genetic components, all localities were dominated by 1. The absence of genetic structure in SNPs inherited parenthood, and the difference in nuclear and mitochondrial genetic structure patterns from other studies indicate possible sex-bias dispersal. This is consistent with the hypothesis that dispersal is mediated mainly by males, as occurs in other elasmobranch species. Sphyrna zygaena, like other highly migratory sharks, uses nursery areas to increase the survival of their young. When taken together with the neutral SNP results from this study, these results strongly suggest that female philopatry is at work. However, due to insufficient sample size for each unit of analysis, it was not possible to conduct tests by sex or size. And on the other hand, adaptive variation from 24 outlier SNP displayed genetic differences between the Gulf of California, represented in orange dots, and the Mexican Pacific coast, represented by green dots. The FST value of 0.061 is low but significant, and the Bayesian cluster analysis indicated the possible existence of two genetic components which may be promoted by individual preferences according to their physiological limitations to dissimilar environmental regions. The SNPs involved in these patterns were blasted to elucidate their function, and they were related to immune response proteins and regulatory elements such as Hox genes and Zinc-finger proteins. In a nutshell, neutral SNP suggested panmixia in the North Mexican Pacific, as well as strong evidence of female philopatry. In the case of outlier SNPs, signal of adaptive selection was detected, and some of those SNPs were related to immune response and regulatory genes. The next step of this is what of these genes are related to environmental conditions. I want to thank to everyone who was involved in this, including the institutions, projects, and researchers, especially to Dr. Axayacatl Rocha Olivares for accepting me in this project during my master's, and Dr. Nancy Saavedra Sotelo, who always encouraged me to do always more than beyond. Thanks for being here. Let me know if you have any questions, preguntas. Hello, my name is Daniela Felix, and I am a PhD student at the University of Manchester. This project was developed during my master's degree.
Okay, now we have time for a couple of questions for Daniela.
So, Daniela, thank you so much for your talk. Super interesting. Studying, as in some of the past cases, organisms that are from Mexico. How do you guys selected these SNPs that you're using, like the 24 SNPs that you figure out that they were under selection or they were important, as well as the 100 and something SNPs before?
For the outlier SNPs, we use two different programs or two different analyses. We use IUCN, that is a Bayesian approach, and use the FST values to detect the SNPs. And also we use the Arlequin, and Arlequin uses also the FST, but also use the island model to detect them. So, we use these two different analyses to detect the SNPs.
Another question? No. I have a question, Daniela. Did you correlate the SNPs that you found like they are like they have adaptation, but did you correlate it with some environmental factors or pathogens that they are in contact with, like with the sharks?
Yeah, thank you. Yeah, actually, it's a good question. We are at that point now. We are trying to find what are the environmental conditions that probably these SNPs are related. But I didn't include at this moment because these are preliminary results. But apparently some of these SNPs are related to some environmental conditions like temperature, chlorophyll, and salinity, because the northern Mexican Pacific region is dissimilar regions. The Gulf of California has a different environmental conditions in contrast to the Pacific. So yeah, we are on that point.
Okay, thank you. Great talk. So now we are going with our next speaker.
Hi.